Our next speaker is my co-moderator, PELIN VOLKAN. The title of her talk is Developmental Programs and Olfactory Receptor Signaling in Structural and Functional Development of Fruitless Positive Olfactory Neurons.
Thank you, and I should tell you that you will see some of the same slides. I also would like to start by thanking the organizers for giving me this opportunity. My lab is interested in understanding how neural circuits get assembled and are modified in different time scales. We're particularly interested in identifying gene regulatory networks that give the structural and functional properties to neural circuits which define their behavioral outputs, and how do processes such as evolution and experience change the transcription or these gene regulatory networks to modify the properties of neural circuits and ultimately modify the behavioral output. So we study this in the context of the Drosophila olfactory system that Scott had just talked about, but just to kind of remind you, there are 50 different classes of ORNs, each class defined by the expression of a single olfactory receptor gene from approximately 80 genes in the genome, and the neurons that express the same receptor basically send their axons, project their axons to the same antennal lobe glomerulus. So obviously olfaction is very important for driving behaviors that are very important for the organism's survival, such as feeding and running away from predators, as well as mating and courtship. And today I'm going to talk mostly about the neurons that mediate courtship behaviors in Drosophila. Oh, no, that's not what I wanted to do. Okay, that's what I wanted to do. Okay, so in flies there are 3 different classes of neurons that have been shown to regulate sex-specific behaviors, and these are the Or47b neurons that reside within this AT4 sensillum, Or67d neurons within AT1, and Ir84a neurons in AC4. So all these 3 neurons detect odors that are associated with driving courtship behaviors. For example, 67D was shown by, these are all work by many labs. So 67D is known to detect cis-vaccinyl acetate, a male-specific pheromone, and it's required for appropriate courtship. So if you knock out 67D, there's an increase in male-male courtship. Or47b detects fatty acids that are fly odors, and 47b was shown to have a role in regulating age-dependent increase in copulation success, and if you mutate 47b, then these older flies cannot mate as effectively as they should. And finally, Ir84a is known for detecting food odors, so the function of Ir84a is basically making sure that there's enough food around to actually initiate courtship, and when you knock this gene out, there's a decrease in courtship behaviors. And of course, with a role in courtship, all these 3 neurons express the critical courtship regulator protein called fruitless. So just to kind of summarize fruitless, so fruitless is a key regulator of sex-specific behaviors. It's a transcription factor that has been shown to regulate the structure and function of the circuit that underlies these behaviors. It was also shown that the transcripts of fruitless from the P1 promoter are alternatively spliced in males and females, and it's the male form that actually generates the functional protein. And if you actually knock this male form out, males start courting other males, and if you knock it into a female, females start courting other females, so fruitless is necessary and sufficient for driving these behaviors. So despite this important role for fruitless in mediating sex-specific behaviors, when you look at its expression profile, it's only expressed in a small set of neurons in the brain, approximately 2,000 neurons. But interestingly, all the FruM-expressing neurons are connected to one another. In other words, if you look at, for example, the olfactory receptor neurons that are fruitless positive, they connect with other fruitless positive second-order neurons, and so on and so forth. So this begs the question, how is the fruitless expression linked to the correct olfactory receptor identities during the development of these ORNs, and are there any effects of olfactory experience on gene expression within these ORNs? Okay. So we started looking at this through a mutant that we identified a number of years ago in a gene called Alhambra, which encodes a chromatin modulatory protein. So Alhambra is required to suppress the 47b and fruitless expression in the other neurons that are housed in the same sensillum with Or47b. So this is that sensillum. So there are normally 3 neurons in here. expressing 47b, 65a, and 88a, and these are the target glomeruli for these three classes of neurons. And what happens in Alhambra mutants is that you basically expand both 47b and fruitless expression to these other neurons at the expense of 65a and 88a expression, but this happens without affecting the general projection patterns of these neurons in the antennal lobe, suggesting that Alhambra is specifically regulating or repressing receptor expression and fruitless expression in these inappropriate neurons. So a lot of these phenotypes I'm not showing you, but they arise in pupal stages during development, which suggests that within the 47b neurons there must be a mechanism that tightly correlates or links the Or47b expression to fruitless expression, either through an unknown factor X or through cross-regulatory interactions between fruitless and 47b. And what happens in these other two neurons is that you have Alhambra, which inhibits either this factor X or simultaneously fruitless and 47b. So this coupling was so tight that we really wanted to investigate this relationship, how this is established. So one of the first things that we looked at is whether there is a relationship between fruitless and 47b. So here, so the first thing that we looked at was fruitless mutants, because fruitless obviously being a transcription factor and critical determinant of the courtship circuit development, we thought perhaps it's regulating Or47b expression. So here you're looking at antennae that are labeled with green for Or47b neurons, and when you look at fruitless mutants, there's no change in 47b expression, suggesting that fruitless does not regulate the expression of this pheromone receptor. However, when we looked at the opposite end, where we are looking now at the loss of function of 47b mutants, we found something very interesting. So here you're looking at, on the top panel, wild-type antennae. So the green labels the fruitless expression and magenta 47b neurons, and you can see all 47b neurons in the wild-type express fruitless. However, when you looked at 47b mutants, what you find is a class-specific loss of fruitless expression only in the 47b neurons. And this is not because the neurons are dying, because we can still see their cell bodies, suggesting that Or47b is required for the FruM expression in Or47b neurons. So Or47b works together with a co-receptor Orco. So we also tested whether Orco mutants give the same phenotype, and they do. So here you're again looking at Orco mutants, and you see that there's a loss of fruitless expression in this zone where normally Or47b neurons reside, and the 47b neurons are still there. Okay. However, one thing that I want you to notice is that there are other neurons, like Or67d neurons, that also require Orco for signaling, and those neurons reside here, and they actually do not have any defects in fruitless expression, and I will come back to this in a few slides. So this loss of fruitless expression in 47b neurons, we can very robustly rescue this by overexpressing Or47b, but we see a very little to no rescue if we overexpress other receptors such as Or88a or Or67d. Okay, so we see this relationship or regulation of fruitless by Or47b, so we wanted to ask if this is true for the other OR genes that are expressed in fruitless positive ORNs. So one of the first ones that we looked at is Ir84a, and so here you're again looking at adult antennae, and these are only labeling fruitless expression in Ir84a neurons, and we are suppressing fruitless expression using an Orco gal-80 in the Or67d and Or47b neurons. And again, if we make mutations in Ir84a, we lose this expression of fruitless in this class of ORNs. So interestingly, when we looked at Or67d, this is not what we see. So Or67d neurons reside in this zone that is depicted over here. And if we look at, again, you're looking at fruitless expression. Again, if you look at Or67d mutants, you can look at them a while, 2 weeks, 3 weeks, and you see that fruitless expression is never disappearing in these neurons. And this is also true for Orco mutants, again, which also works together with Or67d. Again, both Orco mutants and 67d mutants don't have any defects in fruitless expression. Okay. So going back to this relationship between 47b and fruitless, so we know that the onset of both receptor expression and fruitless expression occurs in pupal stages during ORN development or antennal development. So we wanted to ask whether 47b regulation of fruitless is required for the onset during development, onset of transcription during development. Okay, so here we're looking at antennae at late pupal stages, approximately 80 hours after pupal formation. And when you look at fruitless expression in both Or47b and Orco mutants, there seems to be no phenotype, suggesting that the onset of transcription for fruitless is unaffected in these receptor mutants. However, when the flies come out and they start smelling the world in about 5 to 7 days, then we start seeing this loss of fruitless expression in these neurons, suggesting that Or47b function is required for the age-dependent maintenance of fruitless expression in adult Or47b ORNs. So, of course, the next question was whether this requires neural activity, and so far what we've done is overexpress potassium channels such as Cur2.1 or ORC, and in neither case we actually detected any change in fruitless expression, which was very surprising. But of course, if it's not neural activity, then what is it? So we entertained the idea that it might be maybe signaling through these receptors. So we know that odorant receptors in flies are cation channels that allow both sodium and calcium. So we thought perhaps, this calcium influx can lead to activation of a CAM kinase pathway that can eventually lead to changes in the transcription of fruitless locus. So we tested this using RNAi lines against CAM kinase 1 and CAM kinase 2. So with overexpressing CAM kinase 2 RNAi, had a very little to no effect in fruitless expression, but CAM kinase 1 showed a decrease in this zone again that represents 47b neurons, suggesting that it might be going through CAM kinase 1. And so how is CAM kinase 1 doing this? So we know that in mammals, CAM kinase 1 and 4 can lead to phosphorylation of P300 CBP, which was very nicely reviewed in 2 talks before me. So we wanted to ask whether P300 also had an effect in this age-dependent maintenance of fruitless. So we did an RNAi knockdown of P300, and again we saw, this is the gene called Nigiri, again we see a loss of fruitless expression in this domain. Okay. So what I just told you so far today is that there seems to be this 2 different modes of regulation for fruitless expression in the olfactory receptor neurons. First of all, during development, there are mechanisms that couple appropriate receptors to fruitless expression, perhaps either directly, these are unidentified factors, so they're required for the co-expression of these 2 factors in the correct neurons, but then you also have these other proteins that are required to repress both of these identity in inappropriate neurons. And once this pattern is established during development in the adult, some of these olfactory receptors, particularly Or47b and Ir84a, but not 67d, is required for the maintenance in an age-dependent way of fruitless expression in these ORNs. And the signaling through these receptors goes through calcium signaling and chromatin modulatory proteins. So what are the implications of this? First of all, what we're seeing here is basically a compartmentalization of plasticity and stability for fruitless expression in these among these 3 different classes of neurons. So in the case of 67d, receptor signaling does not have any role in regulating fruitless expression. However, for Ir84a and 47b, the fruitless expression requires the olfactory receptor signaling. And of course, one of the things that we're very interested in identifying is that whether this plasticity that's occurring at the molecular level within these neurons is actually a representation of a mediation of more plastic aspects of courtship by these 2 classes of neurons. And with that, I would like to thank the people who did the work. So Catherine, graduate student, and Doug, postdoc, who did most of this work with help from Tristan, and of course people who sent us reagents and money. And also, if you guys are interested in it, so we're also seeing the similar type of compartmentalization across evolutionary timescales within the entire olfactory system. And if you're interested in hearing about the plasticity within evolutionary timescales, please go see my student's poster, D1415A. And thank you. I'll take questions.
We have time for 1 question.